overcome the prevalent problems in clinical decision-making and major depressive disorder and multiple sclerosis, the aim of the CDS-QUAMRI project is the development of a clinical decision support system based on magnetic resonance imaging data. Magnetic resonance imaging provides quantitative information about the anatomy, function, metabolism, as well as microstructure of the human brain. Multiple of these quantitative biomarkers have been shown to correlate with disease progression, therapy response, and can be used for therapy monitoring in both disorders, multiple sclerosis and depression.
Depression is a common and serious mood disorder. At least 350 million people worldwide will live with depression, which makes it the leading cause of disability worldwide. In the European Union, 40 million people suffer from depression. Also, depression is associated with an increased likelihood of severe somatic diseases, and the risk of suicide is about 20 times higher than in the general population. The diagnosis of depression is based on clinical judgment because, unlike in other fields of medicine, there are not yet any biological markers to guide decision-making with regard to diagnosis and treatment selection, which means that selecting the right treatment for a given patient is not yet possible. Rather than the try-and-try-again approach to treatment selection that is currently used, new imaging technologies might facilitate diagnosis and treatment selection by identifying objective neurobiological markers that underlie pathophysiological processes in major depression.
Multiple sclerosis is an immune-mediated inflammatory neurological disease of the central nervous system that attacks myelinated axons, characterized by inflammation, demyelination, and neurodegeneration. Its progression to different subtypes is highly varied and unpredictable, and its causes are still unknown. 2.8 million people are estimated to be affected by MS worldwide, with women being affected 2 to 3 times more often than men, and with higher incidence and higher latitudes. Whether this is due to environmental, genetic, or other factors is still unclear. Diagnosis is based on conventional MRI techniques, and it involves the evidence of damage that is disseminated in time and space and the presence of inflammation. After having ruled out other possible diagnoses, one of MS can be done. However, prediction of clinical progression based on conventional methods is still poor, which is referred to as clinical radiological paradox, hence the need for advanced quantitative MRI techniques.
Magnetic resonance imaging yields complementary information about anatomy, microstructure, function, and metabolism of the human brain. Several related image contrasts show subtle differences in psychiatric and neurological disorders. Hence, the aim of the CDS-QUAMRI project is the development of a clinical decision support system that exploits these subtle imaging biomarkers. To do that, a software is envisioned that integrates analysis modules for structural imaging, functional imaging, and metabolic imaging into a joint framework and allows additional clinical measures to be analyzed as well. Altogether, all these biomarkers will be classified using machine learning-based applications in order to help clinical decision-making with respect to diagnosis, patient stratification with respect to treatment response or disease progression, and monitoring of the treatment response. Let's have a closer look at which information magnetic resonance imaging of the human brain can deliver. First of all, we have anatomical imaging. It investigates the properties of brain water, for instance, its concentration, the so-called proton density, or the exchange of water that is bound or freely moving, the so-called magnetization transfer, and its T1 and T2 relaxation. All of these properties of the brain water are tissue type dependent and help to distinguish gray and white matter. Functional imaging investigates the motion of brain water, mainly via blood flow or diffusion, and it helps us to investigate brain perfusion, the functional brain response up in tasks, or brain networks, structural and functional ones. Finally, metabolic imaging investigates metabolites, compounds, and ions. instead of brain border like the two other modalities. Proton spectroscopic imaging, for instance, visualizes choline, a marker of myelination, glutamate, a marker of neurotransmission, or N-acetyl aspartate, a marker of neuronal health. Complementary, sodium imaging visualizes the concentration of sodium ions, which is the most important electrolyte in the human body and also plays a role in neurotransmission. 31P, so phosphorus spectroscopic imaging, visualizes the pH and enables pH imaging in human brain, as well as metabolite distribution maps of metabolites involved in energy metabolism and myelination in the human brain. Eventually, it is possible to do tracer-based techniques with deuterated or carbon-labeled glucose. This glucose solution can be ingested by drinking, and after some time, the specific labels incorporate into downstream metabolites, and this helps us to draw conclusions about brain energy metabolism. Finally, the clinical decision support system that we envision makes use of all these complementary contrasts and combines them with other clinical markers. And we try to solve different prediction tasks with respect to primary diagnosis, treatment response, and disease progression. In order to do that, we use machine learning analysis. And we tested in this project on two different diseases. One is multiple sclerosis and the other one is major depressive disorder.
I'm Professor Claudia Gandini Wheeler-Kingshott and I am the UCL PI for the CDS-QUAMRI project. Together with my colleague Professor Daniela Alexander and our teams, Drs. Francesco Grussu, Enrico Caden and Antonio Ricciardi, we developed MyRelax, which is a set of tools for relaxometry and myelin imaging that you can find at this GitHub address. We also developed spherical mean technique, which is a clinically feasible microscopic anisotropy index. If you downloaded the software, I hope you enjoy it.
The algorithms that I've developed are implemented in a software package called Lipsia, which is available online, free of charge, and it has been used around the world by many researchers for the analysis of MRI data. At present, clinicians in the hospitals use MRI data primarily visually. They look for brain tumors, for instance. However, in recent years, there's a new opportunity for us now because large international databases have been opened up that contain thousands of brain scans. And these brain scans make it possible now for us to learn about brain diseases that were not accessible to MRI so far. And with new tools, sometimes based on artificial intelligence, we can now hope to make much better use of the content of MRI data.
The Max Planck partner contributes to Metabolic MRI analysis module. It consists out of different components. The first component is MR Spectral, a single-voxel spectroscopy processing pipeline that turns raw data into fully processed spectra that can be input into the next quantification step. The next module is the Profit 1D module, which is a spectral fitting software that models the fully processed spectra and determines the amplitude of the different metabolite peaks and the relative contributions of these peaks. And altogether we can derive millimolar quantification or concentrations of these metabolites out of this information. The third component is a MR spectroscopic imaging, reconstruction and processing pipeline. It consists of several different image reconstruction steps that utilize different acceleration methods and combines them with other processing steps that include, for instance, water and lipid artifact removals. The last component is the MR spectroscopic imaging quantification module. It turns fully pre-processed and reconstructed spectroscopic imaging data that contain a spectrum in each of the imaging voxels into quantitative metabolite distribution maps. To that, a spectral fitting step is combined with several corrections, for instance, for differences in relaxation times, in order to derive metabolite maps in the millimole per kilogram unit, as shown here on the right.
Hello, my name is Matthias van Osch from Leiden University Medical Center, and it is with great pleasure that I present to you the results of this project that we achieved for perfusion imaging. For this, we did not only develop quantification software to allow post-processing of your perfusion data according to the international consensus, but we also improved perfusion imaging and made it more robust for application on the individual subject level. For example, we allowed to actually scan within the same sequence the 4D angiography, shown here on the left side, as well as the perfusion imaging. So getting the whole hemodynamics in one go. Thank you.
The machine learning module is designed to be easily integrated into the software to enable automated decision support. The pipeline is entirely coded in Python using open source libraries. It has been implemented to be applicable on a variety of clinical tasks for multiple MRI modalities, and it is fully reproducible. The ML module is called within the software as a single task, the CDS-QUAMRI predict task. The input required is MRI data in standard NIfTI format. Then the ML pipeline, including processing, modeling, and evaluation, is performed and can be customized by the user. Finally, a prediction, for instance healthy versus sick, is returned as the output. As part of our efforts, we published an open source software library for analyzing brain graphs called Graph Kernels, which is available in the GitHub repository shown here.
The first clinical task for major depression is to distinguish patients affected by major depressive disorder from healthy control. Using multimodal MRI as input, we apply a machine learning classification model. We conclude that fMRI provides a good group separation and interpret the results by identifying relevant activation patterns, as shown in the figure. We also conclude that the multimodal analysis is comparable to the single modality counterpart. In the second task, the goal is to assess patients' treatment response to electroconvulsive therapy. We use structural MRI as input for the machine learning model. With our classification analysis, we can successfully separate responders from non-responders. Furthermore, a regression analysis, as shown in the figure, is conducted to investigate the percentage of symptom reduction, yielding significant correlation.
In one study, we investigated whether a depressed patient could be distinguished from healthy controls using brain activations during a working memory task. Using pattern classification on the brain images, we found that patients could be distinguished from healthy controls with an accuracy of 74%. In another study, we investigated the feasibility of ECT response prediction using structural magnetic resonance imaging data. Again, we applied machine learning techniques to predict individual treatment outcomes. More than half of the patients responded to the treatment, meaning they showed at least a 50% improvement of depressive symptoms. classification of ECT responders and non-responders reached an accuracy of 69%. An additional prediction analysis showed a significant relationship between true and predicted treatment outcomes.
We used a multimodal dataset comprehending relaxometry, diffusion, sodium, and volumetric information. We calculated each metric within different brain tissues and used the data to train a machine learning algorithm able to discriminate between healthy controls and different MS subtypes. Overall, we provided evidence for specific uses of different MRI modalities, relaxometry being particularly meaningful when discriminating MS subtypes, diffusion being an overall marker of macrostructural alterations, total sodium concentration being a marker for subtle physiological alterations, and volume being a good indicator of atrophy in MS. Both relaxometry and volumetric modalities are clinically available. And to further explore the potential of clinically available scans, we used deep learning to extract high-level MTR, which is sensitive to inflammation and demyelination, from qualitative scans acquired routinely. The trained model was able to produce quasi-MTR maps that well correlate with the ground truth, that can be particularly useful when the ground truth is corrupted or missing, giving new life to routine scans in large historical datasets for a more efficient and sustainable research.
The CDS-QUAMRI consortium developed a versatile software framework that allows for multimodal quantitative magnetic resonance imaging in large clinical trials. The next steps include the implementation in the true hospital settings and the extension of our approach to a wide range of psychiatric and neurological disorders.